Moving on to our next topic, which is germination. Till now, we have studied that a seed contains a dormant embryo. Till now, we have discussed that seed contains a dormant embryo. What do you mean by dormant? Dormant is that embryo which is actually inactive. That means it is not at all active to reach a seedling stage, to reach a plant stage. So it is actually at a dormant stage. The first stage of a embryo is dormant, or you can call it, it is the inactive stage of any plant. Now this seed which contains a dormant embryo, whenever it will receive some favorable conditions. So whenever some favorable conditions are achieved during the presence of favorable conditions, I'll tell you what those favorable conditions are. Now, in the presence of favorable conditions, this dormant or inactive embryo, it will convert into the active embryo. It will convert into the active form or active embryo. So, this stage from dormant stage to active stage, from inactive to active stage is known as germination. We call this process as germination. Now, what are the favorable conditions that I am talking about here? The first favorable condition that a plant must have is the presence of water. The second condition is the optimum temperature. A plant or an embryo, it needs water, it needs optimum temperature, that is a suitable temperature, is not very high, not very low. And the third condition that the embryo needs so that it can develop into a seedlet, so that it can actually germinate, is the presence of oxygen. So these three are the most important points that are required for germination, or we can see that these are the favorable conditions that an embryo must have. When these conditions, at optimum water, optimum temperature and presence of oxygen is there, then this dormant stage will be converted into the active stage, or the inactive embryo will be moved into a active embryo stage. So how these three factors are actually important? Let us discuss importance of each of these factors. The first factor that we have discussed is water. So how water is actually important for the development of a seed? Water is needed for two reasons. The first reason is that when water enters the seed, seed will swell. So it will actually develop. This swelling of seed is achieved by the inward movement of the water from the outside. The second thing that happens with the help of water is that enzymes become active. Various enzymes become active, and this activation of enzymes leads to the eating of the food by the embryo, which is actually present inside the seed. So embryo will have those food, have those endosperm food, and then they develop into a seedling. So what happens when water is actually given to a seed? First, the seed will swell, and second reason, the enzymes which are present inside, it becomes active. What happens when these enzymes become active? All the chemical reactions start. So chemical reactions start. That means that it leads to the development of a seed to a seedling, or normally a germination will occur. Moving on to our next step, how you can say that water is necessary? You can conduct an experiment. Now this experiment can be conducted by yourself. Take two conical flasks. This is one conical flask which I am labeling as A. Take another conical flask. Label this conical flask as B. Now what you have to do is just put some cotton inside both these flasks. But this is a wet cotton and this is a dry cotton that we are taking in the part A or in the flask B. The two cotton rolls are taken in the both two flasks and the seed is kept here. These are the two seeds which are kept in both the flasks, both which contains wet cotton and one which contains a dry cotton. After few days, what you will observe that in this flask, in flask A, the seeds start to germinate, which contains a wet cotton, but respectively. In other you can see contrasting that there are no seeds germinating in flask B, however seeds are germinating in flask A. So what is the difference between these two? Only the difference that you maintained was the absence of water and presence of water. You took wet cotton here and you took dry cotton here. So this is the only difference. That means water was necessary for the germination of seeds, but water was absent there. That's why the seeds could not germinate there. So this is the experiment which you can conduct yourself, and this is how you can predict that yes, water is necessary for the germination of the seeds. Moving on to our next experiment, or to our next favorable condition, which is presence of optimum temperature. What do you mean by optimum temperature? Optimum means suitable temperature. Optimum is suitable. How you call optimum? Not very high, not very low temperature is known as optimum temperature. Not very high and not very low. So what is that temperature? It is around 25 to 35 degrees Celsius, not very high and not very low. So at these temperatures, the seed can easily germinate, and this temperature is actually 25 to 35 degree centigrade. So this is the temperature at which is optimum temperature at which seeds can actually germinate. If you take high temperature, embryo will not germinate at all. If you take low temperature, still embryo will not germinate. Why? At low temperature, enzymes are not active, so embryo will not germinate. At high temperature, seed will actually rupture, or embryo can actually get worst, or it can get ruptured by the high temperature, because it will definitely spoil the linings of the seed, so it will not able to be germinate. So these are the two factors which are very necessary for the germination of the seeds. Now how can you prove this, that yes, temperature is necessary for germination of seeds? Again, we will have two flasks. These flasks are covered by a cork. Let us take two flasks and name them as A and B. Again take cotton, but now we'll take wet cotton on both sides. Wet cotton on both sides. Again we have kept some seeds here, some seeds here, and some seeds here. Now, what is the difference that I am making in these two flasks? I want to see whether temperature is actually influencing them. So what I am going to do is, we are going to put this flask, flask B, into refrigerator. Let us put this flask into refrigerator and flask A into normal room temperature. Let us keep this at ordinary room temperature, which is actually 25 to 35 degree Celsius. So ordinary room temperature. I have made a difference of temperature. I have not made a difference of water. I have taken wet cotton on both sides. So water is not the determining factor right now. Now the determining factor is how temperature is able to influence the germination of seeds. In this case, temperature is room temperature. In second case, it is a refrigerator, which is 4 degrees Celsius. So now what we'll observe, that after few days, seeds germinate here, but no germination of seeds occur at this side. So this is again a concluding factor that yes, temperature was a necessary factor determining the germination of seeds. When you take an ordinary temperature, which is the optimum temperature, there was germination of seeds. But whenever you decrease or increase the temperature, you can see that there will be no germination of seeds. So again, temperature is also a determining factor for the germination of seeds, or we can say that it is one of the favorable conditions. Moving on to a third favorable condition which is required for the germination of seeds, which is presence of oxygen. All living organisms then require oxygen for respiration and to carry out various metabolic processes. So again, oxygen is also necessary for plants. So what role does this oxygen has to play? We will have another experiment to prove that yes, oxygen is also necessary. Again, I am taking a conical flask. I'll take two conical flasks. again A and B. Now, I'll take a cotton swab on both sides. This is a wet cotton swab here, and again a wet cotton swab in flask B. This is flask A and this is flask B. I'm taking cotton swab on both sides and I'm putting my seed into all those cotton swabs in two flasks. What is the difference that I'm making in this figure and not in the previous? What is the main factor that I want to see? I want to see that whether oxygen is actually influencing the germination of seeds or not. So what I'll do, I'll keep a test tube here and I'll keep another test tube here. And I'll hang this test tube from this cork. Again, I'll hang this test tube from this cork with a thread. Now, in flask A, what I'm going to do is I'm going to put plain water into this flask. I'm going to put plain water into this test tube, and into this test tube I'm going to put pyrogallic acid. What is pyrogallic acid? Pyrogallic acid actually absorbs all the oxygen. So it will remove whatever oxygen is present in the flask, it will actually remove all the oxygen. I'm putting pyrogallic acid here, I'm putting plain water here. Now what you can see is that oxygen is absent in this case because I put pyrogallic acid, which is actually removing all the oxygen. So oxygen is removed in flask 2, but oxygen is present in flask 1. Now what do you see after a few days? That seeds germinate in flask A, but they do not germinate in flask B. This means that oxygen was present here and oxygen was not present here. So non-presence of oxygen does not germinate the seeds, but oxygen presence is needed so that germination of seeds can actually take place. So this leads to our third point, that yes, for germination oxygen is also required. So these are all the favorable conditions that are required for the germination of seeds.